Welcome to this episode. Today we're diving into what makes the Neurophorol Xanthene core concept fundamentally different from traditional cannabinoid scaffolds. It's a fascinating shift in molecular architecture that really changes how we think about cannabinoid research.
Yeah, it's a great topic. Last time we talked about why scaffold design matters, but this time we're going deeper. Looking at how changing the core structure itself, not just tweaking the edges, can reshape the entire research framework.
Exactly. Traditional cannabinoid research often builds on well-established frameworks, scaffolds that have been around for decades. But Neurophorol challenges that by rethinking the core architecture itself. It's like redesigning the molecular skeleton, not just redecorating the surface.
Right. And that's a big deal because the core scaffold governs so many fundamental properties: geometry, planarity, how substituents are presented, and ultimately how the molecule interacts with biological targets.
When you change the core, you're not just changing the molecule's look. You're changing its three-dimensional shape, how flat or twisted it is, and how it fits into receptor sites. That can completely alter selectivity, meaning which targets the molecule prefers or avoids.
And that's crucial for medicinal chemistry, isn't it? Because selectivity drives therapeutic specificity. So a different core scaffold can open up new interaction patterns that we wouldn't see with traditional cannabinoid frameworks.
Yeah, and it forces medicinal chemists to rethink structure-activity relationships. The usual SAR paradigms might not apply directly when the core architecture shifts. So instead of just incremental tweaks, you get fresh avenues for exploration.
That's a really important point. Sometimes novelty is just novelty for novelty's sake. But here, the structural innovation is meaningful because it reframes the scientific conversation. It's not just about making a new molecule. It's about asking better questions.
Exactly. Neurophorol isn't just a name or a new compound. It's a structural idea that changes how researchers think about selectivity, mechanism of action, and medicinal chemistry decisions from the ground up.
I like to think of it like architecture. Traditional cannabinoid scaffolds are like classic building designs. Changing the paint or furniture is surface level. But Neurophorol is redesigning the foundation and frame of the building itself. And when you change the foundation, everything else changes. How rooms connect, how the building functions, what you can do with the space. Similarly, changing the core scaffold changes how the molecule behaves and what biological interactions become possible.
That analogy really helps visualize why this architectural shift matters so much. It's not just a cosmetic change. It's a fundamental redesign that opens new possibilities in drug discovery.
And in early-stage biotech, that's exactly what you want. A platform defined by the quality of its scientific questions, not just by rushing to outcomes or chasing novelty.
Right. Neurophorol exemplifies mechanism-first thinking and rigorous validation. By focusing on architectural innovation, it sets the stage for more targeted, selective, cannabinoid-inspired therapeutics.
So, to sum up, the Neurophorol Xanthene core concept changes the entire architectural conversation. Geometry, planarity, substituent orientation, and selectivity. That leads to better scientific questions and more meaningful medicinal chemistry decisions.
And ultimately, it reminds us that in research-stage biotech, the value of a platform lies in the rigor of its framework and the quality of its questions, not just in novelty or immediate outcomes.
Exactly. Neurophorol embodies that principle perfectly. Mechanism first, validation always.
Well said. Thanks for joining us for this deep dive into the Xanthene core concept. We hope it gives you a fresh perspective on scaffold design and cannabinoid research.
Yeah, thanks for listening. Until next time.